Great. Thanks so much, Tanya, for that introduction. And good morning and welcome to everyone joining us today for the webinar. Before we get started, I just wanted to make a couple housekeeping announcements. So as Tanya mentioned, for today's webinar, I'm going to give a introduction to a number of different preclinical modalities that are commonly used in imaging research. And while all of the example images that we'll show today were acquired on imaging systems that we distribute here at Scintica, for the purpose of today's session, we won't actually go into any details regarding the products themselves. So this is going to be more of an educational session to get everyone up to speed. However, I did want to mention that we are running our preclinical imaging series in the next 2 weeks. And so if you're already very familiar with preclinical imaging, I do encourage you to join us for these other events. The first is going to be next week on October 1st. This is an all-day event where we'll be doing live demos of our bioluminescence, ultrasound, and MRI systems using various mouse models. And if you're only interested in one or two of the systems, you're welcome to come in and out of the event during the day. But because it's a live demonstration, it can't be recorded. So you do have to attend the event live. And then on October 6th, we're going to be doing a 60-minute webinar. And this is where we'll actually discuss the specific technical capabilities of all of our preclinical imaging products here at Scintica. We'll also show some of the images that were taken during the live demonstration. And so if you haven't already, feel free to go to the Scintica website that I've provided here at the bottom, and you'll find the link to register. Okay, so for today's webinar, I'm going to spend the first few slides talking about the need for preclinical imaging within research. We'll then move on to introduce each modality we'll talk about today. So as Tanya mentioned, this will include MRI, PET imaging, ultrasound, optical imaging, including both bioluminescence and fluorescence. And then AS is going to take over for a bit to teach us about endomicroscopy. We're going to give a very brief technical overview of each modality. We'll discuss some of the strengths and weaknesses of each system, as well as go over some of the common applications, so you can start to understand what different things you can study with each of these modalities. And then near the end, we're also going to cover something called multiplex imaging, so what that means, and then when and where you may want to consider this in your research studies. Okay, so jumping right in. So the field of preclinical imaging really grew out of the enormous wealth of information that's provided by in vitro diagnostics. So as many of us know, these assays can include things like histological assays, protein assays, microarrays, and these all really give us information regarding the changes that occur during the pathogenesis of a disease. However, these assays really do have some limitations. Importantly, they only sample a part of the subject or disease tissue. They give us static information, so at only a single time point during the disease process. And they're invasive and typically destructive, which limits the amount of information we can really get out of that sample. And so these limitations led researchers to envision whether it's possible to perform these types of assays in living subjects. And so we now know this as in vivo imaging or imaging assays. And so here the advantages are that you can assay the entire subject. You get dynamic information since you can image the same subject over time. And for the most part, these assays are non-invasive. So this leaves the underlying biology intact so that we can track changes in the disease process. And so overall imaging provides a more complete picture of the disease process or the biological phenomenon that we want to investigate. And so preclinical imaging modalities have really become crucial for scientists to observe these changes, either at the organ, tissue, cell, or even molecular level. In most cases, functional changes will occur much earlier than structural changes. And so non-invasive imaging has really enabled this longitudinal investigation to better understand that natural progression of disease. Now, there's a wide variety of preclinical imaging tools to choose from. And really, there's a whole field now that's dedicated to instrumentation and technology development. And so this list continues to grow. But for the sake of today's webinar, we'll be focused on 4 of the most commonly used modalities in preclinical research. And again, this is positron emission tomography or PET, magnetic resonance imaging or MRI, optical imaging, and then finally high frequency ultrasound imaging. So we're going to start with MRI. So for those who are very familiar with MRI, please just bear with us as We do really want this to be an educational session, getting everyone up to speed and familiar with all of the modalities. So while all MRI systems can be a little different, in general, an MRI scanner is comprised of a set of embedded coils. So we have one coil that generates the main magnetic field, which we see here in orange. We have gradient coils that are used to localize the source of the MR signal. And then finally, we have RF coils shown here in yellow, which excite the sample and detect the resulting MR signal. And so the magnetic field from the MRI will interact with certain atomic nuclei, either in the body or elsewhere, that have special spin characteristics and magnetic properties. And this is most often, we're using hydrogen atoms since they're abundant in living subjects, particularly in water and fat. And so usually these protons are facing in random directions. But when a subject is placed inside the magnet, a significant portion of them will align in the same direction. And then a radio pulse is emitted, which interacts with the protons, essentially turning them so that they no longer align with the magnetic field. But this only happens for a moment before the protons will relax back to their aligned state. And as those protons relax, energy is released. And this is actually what's detected by the sensors in the MRI. And then through some calculations, the computer can determine what the tissue looked like depending on this energy that's released. Now, by changing the timing of that RF pulse, collectively known as a pulse sequence, so some of us may have heard that term before, different contrasts can be generated based on the relaxation times of different tissues. So for example, here we have a T1 and T2 weighted image of the same mouse body. And so in a T1 weighted image, fat appears bright, and in a T2 weighted image, water is going to be bright. And so we can see here just from looking at these images that based on the parameters you modify or the pulse sequence you choose, and these are just two examples, you can highlight different structures throughout the body. MRI has a number of important advantages compared to other imaging modalities that we'll talk about later. One of the most valuable being the unlimited depth of penetration. So you really can image essentially any tissue within the body. It has high spatial resolution. It's very quantitative, so concurrent collection of physiological or metabolic data is possible. Another important advantage is that it doesn't use ionizing radiation. And so this becomes important when we start to think about longitudinal imaging or treatment studies where we want to image the same subject many times in a short time frame. And then not always important, but definitely something to consider, especially for those writing grants, is that MRI is obviously very clinically relevant. And so this can help to speed things along that translational pipeline compared to a technology that's exclusively preclinical. So moving on to limitations, and as I go through these for each technology, I want to consider these more, I like to call them more considerations, just because they can be overcome. It just really depends on your specific application. And so the first limitation of MRI is poor sensitivity compared with other imaging modalities. And this low sensitivity can lead to relatively long acquisition times. And in the case of molecular MR, it's the reason why large amounts of imaging agents are often required to obtain an adequate signal. And again, this totally depends on the probe that you're using. Additionally, many of us know that MRI is very expensive, not only for the system itself, but it can be very costly to both maintain, as well as set up in terms of having the proper infrastructure. And then finally, it has relatively poor temporal resolution compared to other imaging modalities. And again, this relates to that longer acquisition time. So MRI can be used for a wide variety of applications, including anatomical, functional, and even molecular imaging, as I mentioned. I would say the most common types of studies typically include neuro, oncology, cell tracking, as well as cardiovascular studies. It's also commonly used for ex vivo imaging for things like MR-based histology, and this has to do with that really nice high resolution. So we'll just go through a couple examples. So I would say one of the most common applications would be neurology, both due to the superior soft tissue contrast that MRI offers, as well as the limitless depth of penetration. And so if we compare it to another modality, for example, optical, which we'll talk about in further slides, The brain can be very difficult to image just because in the case of optical, signal relies on light getting through various structures such as the skull. Whereas with MRI, by adjusting those different parameters, you can really highlight nearly any structure within the brain. And so in this case here, we can see a lesion in the brain following a stroke, and it appears bright due to inflammation and likely fluid in that area. Here we see images of the brain again. This time it is a preclinical tumor model. And so 15 days following cell injection, we can see a tumor appearing bright, again, likely due to fluid since this is a T2 weighted image. Another common application for MRI is cardiovascular studies. So an example is angiography, and this is used to generate images of arteries and sometimes veins in order to evaluate them for things like stenosis, occlusions, aneurysms, or other abnormalities. And this is often done with a contrast agent such as gadolinium. Okay, so this was a whirlwind, I know. We're just covering a very few applications, but keep in mind there are many more, and we will go through a couple new applications both next week in our live demo, as well as the following week, showing example images in our product overview. If you do have a specific application in mind that you have questions about, feel free to reach out to us and we can address those questions for you. Okay, so moving on now to positron emission tomography or PET imaging. So PET imaging is something some of the non-imagers may be a little bit more familiar with due to the fact that it's routinely used clinically to diagnose and monitor the progression of numerous diseases, including a wide variety of cancers. But it's also commonly used in many areas of research. And so to image a certain molecular target using PET, a radio-labeled imaging agent is administered to the subject. Typically, this is done through an intravenous injection. And the imaging agent can be really anything, a small molecule, a peptide, a nanoparticle, etc. But it needs to be specific and selective for the target of interest. And what happens following that injection is radioisotopes will decay. And as they decay, they emit a positron, which will then travel a short distance and collide with an electron in the surrounding tissue. And this interaction causes the release of 2 gamma photons in approximately opposite directions. Now, following the detection of gamma rays by the PET detector, which we can't see in this schematic, but it would be surrounding the animal here, the location of annihilation events are calculated by observing multiple events. And so the resulting electrical signals are then converted and reconstructed into a tomographic image. And then these images would reflect the distribution of the imaging agent in the subject. And so this provides information, of course, on the biochemical event that the agent was targeting. And so I'll just show an example because it's easier to understand with an image. So here we have an image where the animal was injected with FDG, which is just a radioactive version of glucose. And so we're able to image the metabolic activity within this tumor, which I don't think in this case I need to point out for anybody. But we can also see other structures in the rat, such as the myocardium and the brain. So since biochemical changes generally occur before anatomical changes in disease, as I mentioned earlier, PET does have a clear diagnostic advantage over some of those anatomical techniques such as CT and MRI. I would say the key strengths of PET include its excellent sensitivity, that limitless depth of penetration, similar to MRI, and quantitation capabilities. And then similar to MRI, of course, it is also a clinically relevant technology. However, it is quite costly and it does require radioisotopes. And so this is something that needs to be considered. Specialized equipment aside from the system are often needed, such as a cyclotron. And compared to MRI, PET also uses ionizing radiation. And so the dose of radiation during each scan, as well as how many scans would be required during the length of a study, need to be considered. And then additionally, the spatial resolution of PET is a bit limited compared to that of MRI. So traditionally in preclinical imaging, researchers from oncology, neurology, cardiology, and many others have found benefit in using PET to better understand a variety of molecular targets or to study the disease progression. Additionally, PET's been used to study the biodistribution of specific targets, as well as therapeutic compounds. And dynamic imaging is starting to become really popular to study more of the kinetics of tracer migration throughout the animal model. So we'll cover just a couple examples again. So I would say the most common application of PET still remains to be oncology. And so in this example, the mouse had two tumors, one that was positive for PSMA, or prostate-specific membrane antigen, and the other that was negative for the antigen. And so in this experiment, the PET agent was specifically designed to target PSMA, and we can see, we can nicely visualize that positive tumor over time. We also can see that this agent does have some affinity for the liver, kidney, and bladder. PET can also be used to study inflammation. So here, FDG was used to look at metabolically active areas in a rat model of arthritis. So this is a pretty cool application, and we can see that we don't see this metabolic activity in the control animal, so it's quite a large difference. Another common application is cardiovascular studies. And so in this example, the rat was injected with FDG to look at the metabolic activity of the myocardium. And so in these images, we can see both the left and the right ventricle and the difference in metabolic activity between these walls. And we can actually also see some of the small structures in the left ventricle called the papillary muscles. And then the final example I'll give is bone studies. And so in this case, they've used sodium fluoride as the radio tracer, and it's injected into the rat, and it's taken up in the bones over time. And so these types of images can be used to study things like bone cancers, metabolic bone diseases, or even rheumatoid arthritis. Okay, so moving on to ultrasound imaging. So ultrasound, it's an imaging tool that exploits the properties and behavior of high frequency sound waves as they travel through that tissue. And so if we want to think about using something like microbubbles, so a targeted agent, what would happen is following the administration of the microbubbles, a transducer, which is also called a probe, converts electrical signals to ultrasound waves, and the ultrasound waves will enter the body, and then some sound waves are reflected back to the transducer, where they're actually detected and then converted into electrical signals. And it's these signals that are processed by the computer and then displayed as an image. Now, this is just the case for using something like targeted microbubbles for molecular imaging, but we can actually image without an imaging agent in this case, and we can see some images here of the normal mouse body. And depending on how deep the tissue is that you're interested in, you can see quite a bit. So here we have the skeletal muscle, we can see lymph nodes, liver, spleen, and even cysts and ovaries. So there's quite a wide application of studies here. So the main strengths of ultrasound include its relative cost effectiveness, especially compared to the modalities we've mentioned already. It's widely available, and it's more portable than these other systems. It has good temporal resolution. It has a good safety profile, so there's no ionizing radiation. It also has excellent sensitivity, especially when microbubbles are used. Now, on the other hand, ultrasound is limited by its difficulty in imaging structures that contain bone or air. And this is due to the tendency of bone and air not to transmit sound waves, and also by its limited depth of penetration. Other limitations of ultrasound include the fact that it's primarily anatomical information. And so this really depends on the application that you're interested in. And then multiplexing is not currently possible, and we'll discuss multiplexing in a couple slides. The main applications of preclinical ultrasound include oncology, so things like tumor detection, studying the tumor vasculature, or visualizing tumor-targeted therapies. A wide variety of cardiovascular applications, image-guided needle injections, and tracking things like drug delivery. So here's an example where a tumor-targeted theranostic agent was created. And this study very nicely highlights the use of ultrasound to image over time because it is relatively cost effective, and also it's quite easy to get that same image over time. So here we see the pre-injection where there's no contrast, and then over time we see within that same tumor, we can see the clear accumulation of those targeted nanobubbles. In cancer biology, ultrasound is commonly used to detect tumors, and you can quantify tumor progression over time by doing things like 3D tumor volume measurements. And this can be done both for primary tumors as well as for METs, depending on where those METs are located. But as I mentioned, a common application I would say is METs of the lymph nodes. Lots of groups are also using preclinical ultrasound for cardiovascular studies. So for example, to image vasculature. So here we see a really nice example of the rat carotid artery. And we can get measurements from these images for things like wall thickness. We can monitor the formation of plaques or even do studies looking at vessel dilation over time. And then the final example I'll show is in regards to image-guided injection. So this can also be done with ultrasound. In this case, you would have an anesthetized mouse, so we can actually visualize the beating mouse heart in real time. And then you can see both the needle tip as it's retracted and inserted into the mouse. And then in the case where you could use microbubbles, you would actually be able to see contrast following the injection of your cells or drug, etc. Okay, jumping into optical imaging. Now for optical imaging, as I mentioned, I'm going to go over both fluorescence and bioluminescence imaging. They are quite similar, but there's some very important differences that I would like to cover. So we'll start with fluorescence. So for fluorescence, following the administration of a fluorescent imaging agent, so this can either be a fluorophore or a fluorescent protein, the excitation light of appropriate wavelength is used to illuminate the subject. In the case of using a protein such as GFP, you do need to consider that you would need to first engineer the cell line of interest to express GFP, or you could purchase a transgenic animal that already expresses that gene of interest, and then you would illuminate the subject. Now this leads to the excitation of the fluorophore or the fluorescent protein, and then the subsequent emission of light. And then this light gets collected by the CCD camera, and it gets converted into an image which would detail the location of the emitted light. And so just to give you an example here of an image, this mouse had a subcutaneous tumor that was expressing M-cherry. And so for those of you who are familiar with fluorescence, you may know that this isn't really what I would call a typical fluorescence image, because fluorescence, we do get quite a bit of autofluorescence often. But just as a rule of thumb, the further we go into the red reporters or the infrared, you do have better sensitivity. And so you can reduce quite a bit of that autofluorescence. Overall, I would say fluorescence imaging is very versatile. So it does have a number of key advantages, including its relative low cost and the fact that it doesn't require the administration of a substrate like bioluminescence imaging does, which we'll talk about next. It does serve as a useful tool for preclinical evaluation of imaging agents prior to moving them into other more expensive modalities like MRI or PET. And I would say one of the most important strengths of fluorescence imaging is that it enables multiplexed imaging, which gives you a lot more options in terms of how you study your disease. There are a number of limitations that need to be considered. So definitely the limited depth of penetration, which is typically about 1 centimeter for fluorescence. The mass amount of fluorescent agent needed for imaging is often considerably larger than for something like radionuclide imaging. Furthermore, it suffers from issues associated with autofluorescence, as I mentioned, and this ultimately decreases the sensitivity of the technique. But there are a number of strategies that can improve the overall sensitivity. So including methods that correct for autofluorescence, the type of imaging agent selected. So as I mentioned, the further red you go, the better sensitivity you'll get. And also the type of imaging instrumentation used. So these are all things that need to be considered when planning out your study. Okay, so now we'll jump into bioluminescence imaging. So similar to fluorescence, bioluminescence has really been adopted by a lot of researchers as a valuable cell tracking tool, specifically in cancer research, but it really has an endless amount of applications. For bioluminescence imaging, first, the small animal model needs to be developed that's capable of expressing or secreting one of the luciferase enzymes. And so again, this could be a transgenic mouse or a mouse that's been injected with cells that can either express or excrete luciferase. Now, the first difference between fluorescence and bioluminescence is that in this case, we now need the appropriate substrate that needs to be administered to the animal. This is what's going to lead to the production of light. And so it's actually this enzymatic reaction between the luciferase and the appropriate substrate that leads to the emitted light. And from here, it's the same as fluorescence, where that light gets collected by the CCD camera, and we get an image that's representative of where those cells are. And so here we have an image, and this is an image very similar to fluorescence. We can see here we have, this is a model of orthotopic lung mets that were expressing luciferase. So one of the main advantages of bioluminescence imaging compared to fluorescence is, as I mentioned, the higher sensitivity. And this is just due to the low background signal because obviously there's a lack of natural bioluminescence in the body. So that really helps with sensitivity. It's relatively cost effective and it's extremely user friendly. So I would say both bioluminescence and fluorescence are probably the best option for any non-imagers that are looking to add an imaging component to their lab. And finally, BLI allows for multiplexing, similar to fluorescence imaging, and this is just done by combining different luciferase substrate pairs. Now, bioluminescence imaging does have a limited depth of penetration, just like fluorescence, but the primary limitation is getting light out of the subject, whereas with fluorescence, it's limited by both the ability to pass light in and out of the subject. Depending on which luciferase you use, a substrate and other cofactors such as ATP and oxygen are required to get signal. And so this can either be a limitation or an advantage. I like to think of it as an advantage, just depending on what you are studying, because you're only going to get signal from live tissue in this case. And so if you think of a study looking at the treatment of a tumor, if you use something like MRI, it may take, a month or two for the size of the tumor or the volume of the tumor to look any different in MRI. But if your treatment is working, you should see those differences right away in BLI because within that tumor volume, you're going to have both live tissue and dead tissue. However, do keep in mind that this is a preclinical technology only. Okay, so as I mentioned, there are a wide variety of applications for optical imaging. It really can be applied to almost any study. I would say the most common are oncology, neuro, biodistribution studies because you're getting that whole body visualization, treatment studies specifically for bioluminescence, cell tracking, and then also ex vivo imaging. So this is an example of fluorescence imaging. And this group was interested in studying the potential mechanisms used to enhance the bioavailability of doxorubicin hydrochloride. And so in this study, rats were either given dox-loaded nanogels with or without calcium. And for the sake of this webinar, I don't really want to focus on what those two different conditions mean, but rather the information we're getting. And so you can see really nicely, we can see the whole body. And because this is a biodistribution study, this group may have not known where these nanogels were going to go. And so it's a really great way to get that whole body visualization. And it's very easy to compare one condition to the other. We do notice that because it's fluorescence, we do have some issues with autofluorescence, especially in the fur. And so what's commonly done in fluorescence is just to better localize some of this signal because it is a 2D signal. Many groups will typically do ex vivo imaging after. And so in this case, they wanted to get a better idea of where exactly the signal they were seeing in vivo was coming from. And so after sacrificing the animals, they took out all of the organs and imaged again. And you can see you get a much better idea of where that signal is coming from in each individual organ. Here we have an example of bioluminescence imaging. So this group established a mouse model of peritoneal metastasis using xenografts that stably express luciferase. They then administered either PBS as the control or a number of different treatment combinations, and then used BLI to monitor tumor growth. And again, for the sake of today's talk, we don't really care about these different treatments, but rather the information you can get from using BLI for this type of treatment study. And so again, we can see how easy it is to compare all of these different groups across the board. BLI is extremely rapid throughput, and so most systems typically allow you to image 5 mice at a time, which is really nice. We can also see how important it is to image over time for these types of studies. And so if I pulled the data from week 3, I would get very different results than if I had only imaged at week 7. And so this is something to consider, you know, for more exploratory studies, especially again, because you're getting that whole body visualization. Not so important if you have a very localized signal, maybe you have a subcutaneous tumor and you know exactly where it's going to be. Then you could consider, you know, other modalities that are more expensive, maybe MRI. But in this case, if you want to know throughout the whole body where your signal is coming from, BLI, I would say, is a great place to start. Okay, so just quickly, I want to go over multiplex imaging because I have mentioned that a few times now. So multiplex imaging is the simultaneous acquisition of multiple signals, and these signals are transmitted through a shared medium and then subsequently demultiplexed. And so the modalities that I've mentioned so far that have these capabilities include bioluminescence, fluorescence imaging, and then less commonly also PET imaging. And so here we have an example with fluorescence imaging where we have simultaneous monitoring of red and green fluorescent signals by dual color in vivo imaging. And so these signals can be overlaid so that several reporters can be visualized simultaneously. Now, many people, when they first hear about this, often wonder, well, why would I need 2 different reporters? And there's actually quite a few applications where this can be really useful. Especially if you're not in an imaging core and you really only have maybe one imaging system to work with. So I'd say the most common application is cell tracking. So I've seen many people do different cancer cell lines. So you could maybe tag a primary cancer cell line with red and a metastatic variant with green, and then see how those 2 cell lines interact within the same body. Another common application is cancer cells and therapeutic cells. So being able to monitor, you know, the progression of disease with one reporter while also tracking your therapeutic with another. And again, this would be using the same modality. So that can be really, really cost effective. The second one is immunology. So a big one would be the CAR T cell group. So sometimes they want to separate out their T cell population. So for example, CD4 versus CD8 T cells and seeing how those are contributing to a treatment effect. And then finally, neurology, because we know the brain has very complex biology. So having the opportunity to, you know, track more than one population of, for example, stem cells can be really helpful. And then, as I mentioned, less commonly, PET imaging. And so in this example, FDG was injected to look at cerebral metabolic activity, while an iodine tracer was injected to study dopamine transporters. And so what happens is the combined multiplex PET image is co-registered with the CT, and then the software can actually separate out the various signals. And so how this works, as I mentioned earlier when we were talking about PET, for standard PET isotopes, they routinely emit 2 gamma rays during an annihilation event, but there's also some non-standard PET isotopes that emit 3 gamma rays. And so depending on the system you're working with, you can actually separate out these signals to monitor 2 different biological processes at the same time. Okay, so now I'm going to hand it off to Ayaz, and he's going to teach us about endomicroscopy.
Thank you so much, Katie. And thank you, Tonia, for the nice introduction. So I used to work with Scintica as a product manager in 2018-19. And during that time, I worked with this fluorescence in vivo endomicroscopy technique. And I loved it so much, actually. I'm still associated with Scintica whenever they want to do any educational show. I'm so happy talking about this technology. So basically, Katie talked about the in vivo and ex vivo techniques like MRI, CT, and PET, which are non-invasive, and they give you the idea of what is the disease progression and what's happening inside the body of the animal. But as a biologist, I know like I need to look at the cells as well to understand like what's happening at the cellular level. And the option I have is the benchtop microscopes, and if I need like really good resolution, I have to go for the confocal microscopes. But the disadvantage there is they are highly invasive. So you have to sacrifice the animal, take the tissue out, process slides. So you have two worlds. Basically, you have the microscopy world where you have really good resolution, but then it's highly invasive. And then you have the in vivo imaging systems like MRI and CT, where you don't get the resolution, but it's highly like you can image the live animals. So what this fluorescence in vivo endomicroscopy technique is, it's basically combining the best of both the worlds. So it is a probe-based system, and it's a confocal microscope. So what it does is that the probe goes inside the body of the animal, and then you can capture the images with cellular resolution. So it's minimally invasive, but it still gives you resolution in micrometer scale. And it's a very simple tool, so you don't need to have like really high-end facility, or you have to have a dedicated technician. So anyone who is working with, who has some basic training in dealing with animals can use this microscope. Even an undergraduate student can use this instrument. And it's basically a probe that you have to put inside the body of the animal, and you will start capturing images. And you get live images from the animals. For example, here we injected sodium fluorescent, and you can see how the blood is flowing around the red brain glioblastoma. So this shows how the technology works. So it's basically a confocal microscope, and it has a 488 nanometer laser. That laser is pumped into the optical fiber and into the probe. So this probe is about 3.8 millimeter in size, and then it goes inside the body of the animal. And then the fluorophores are excited, and the resulting fluorescence is collected back. And you can there is an option of selecting between 12 different filters, and anything that's emitting above 515 can be collected. And because it has a confocal mechanism, anything that's above or below the imaging plane, it is removed, the signal is removed, and you get really sharp and precise images. And there is also a scanning mechanism, so it gives you like a field of view of 475 by 475 micron, and it also has the capacity of doing optical sectioning, capable of going up till 400 micrometers inside the tissue. So just to show you how easy this tool is, when I was working as a product manager, I did not have the permission to handle any animal or the ex vivo tissues. So what I did was I took a piece of onion and put down some acryflavine, washed it, and this is the probe of the instrument. So I bring it in touch with that, and you can see the blue laser dot, which is the excitation. And then this is the interface of the instrument. So you start the laser, and I'm knowing like acryflavine will be staining the nucleus. So I'm hoping to see the nucleus. But when I switch on the instrument, I was surprised, like it's not the nucleus. I also get to see the cell wall or the cell membrane. So somehow acryflavine is also localizing into that. And when you capture the images, you can see the structure of the onion cell nucleus, onion cell wall. If you zoom in, you can see the nucleus, and even you can see the nucleolus. So it's a tool which is capable of capturing images live. So it's a clinical as well as preclinical tool. So in clinical setting, it is used for identifying the tumors during the surgeries. So here you can see the 5-2 images with cancer tissue. Sorry. And you can see the breast cancer tissue. And these images are like instant and live from the surgery. While if you go to the classical histology, you have to take out the tissue, send it for pathology, and you'll be getting results like 3 to 7 days later. So in clinical setting, it is used for dissecting out the tumors. In preclinical setting, there are even hundreds of applications. So the basic application is you can do the microvascular imaging and cell tracking. So in the left, in this image, you can see the blood vessels of the mouse brain. And what was done was sodium fluorescence was injected, and you can see the nice architecture of the blood vessels because of the flow of sodium fluorescence. And after sodium fluorescence, acryflavin was injected intravenously. So acryflavin goes and binds to the nucleus of smooth muscle cells. So you can see the organization of arteries, and you can also see some blood cells moving around into this blood vessels. And then a kind of disturbance was introduced into this vessels. So you can see the blood clotting and damage to the blood vessels. So this way, depending upon what kind of application you have, you can do the blood vessel imaging. You can also do simple cellular imaging by the topical application of fluorescent. So here dog stomach was sprayed with fluorescent, and you can see the nice architecture of the dog stomach. Similarly, dog intestine was sprayed with acryflavin, and as we know, acryflavin binds with the nucleus. So you can see the nice architecture of the tissue. You can see the breast border and the mucus layer. We can also see some cells ejecting out of the epithelium, and we can also see the cells dividing, the cells undergoing division. And this were all captured from live animal. You can also do optical sectioning. So here, this is an example of optical sectioning. So lymphatic vessels were stained with the fluorophore tagged antibody. And once you have taken that optical sectioning, you can take it to softwares like IMARIS or ImageJ, or if you have any other in-house software for viewing the optical sections. And you can see the nice architecture of the tissue as well as the organization of the lymphatic vessel. So this is like, there are hundreds of applications that we can discuss about, but because of the issue of time, these are just the few of the images that and my experience with the instrument that I shared.
Great,
Back
thanks,
to you,
Sayed.
Kitty.
Now, I'm going to jump in just for a quick second. Thanks, Sayed and Katie, both for the insightful discussion so far. Before we turn things back over to Katie, I would like to take a quick second to launch our final poll for the webinar today, and just reaching out to try to see if there's any of the imaging modalities that either Katie or Ez have spoken about today that you'd like a little bit more information about the products that Scintica Instrumentation does offer. Again, the images that both of them showed are from products that we offer, although today hopefully we're just discussing the modalities themselves. I'd also like to take this opportunity to have you use the question and answer dialog box to ask any questions that you may have thought of throughout the webinar. And again, once Katie's finished a quick summary, we will delve into those and answer some of the questions that you guys may have. So I'll give everyone a couple more seconds just to respond to the poll. Wonderful. Thanks again for taking the time to answer the poll question. And at this point, I'll turn things back over to Katie to finish up the presentation.
Great. Thanks, Tanya. Now, just to finish up, I promised you we're going to get you out in time for lunch. But just to summarize, I wanted to go over a couple things to consider when choosing a modality for a study. And so these obviously aren't all of the questions I would ask, but this is a good place to start. And so the first one that comes up often is, do you need to image over time? And so this is important because
I mean, for some of you, you may just want an endpoint measurement. So let's say you're doing a tumor study and you may just want the most complete data set at that one time point. And in that case, you may want to consider something like MRI, where you can get like a volume measurement. If you are going to be imaging multiple times, you may want to consider something that's more cost effective. So something like optical or ultrasound imaging. Do you have the ability to engineer cells? So again, this doesn't always have to be a question you ask, but if you are considering something like optical, you might want to make sure that either you have either transgenic animals available or you're able to engineer the cells with the gene of interest. How deep is the tissue of interest? And so again, this isn't always important. So for groups that are looking at maybe subcutaneous tumors, you really, your options are unlimited. But if you're looking at something that may be in the brain or the lungs, you might want to consider one of the modalities we mentioned that has unlimited depth of penetration. So for example, MRI, PET, or CT. Do you need quantitative data? And I always like to say, how quantitative do you need? Because quantitative, of course, means a lot of things. And this can be different for each modality. So whether you need, an exact cell number, so how many cells have got to a specific location, versus maybe you just care about the relative difference in signal between groups. And then finally, I would say probably one of the most common questions is, do you need functional or anatomical information? And I would say more commonly than not, this is quickly becoming, you need functional and anatomical information, or people would prefer that to get that complete picture. And so a lot of our systems have started to become integrated. So we have options for PET CT or PET MR in this case. And I just want to leave everyone with this resource. So I covered quite a few of these modalities today, but like I said, there are many others. And this is a really great resource, both for those of you who are just getting into imaging and want to learn more about it, but also for those who have been imaging for years. This is a really great review article, and it's a great place to start if you're thinking about, diving into an imaging study and you want to know for your specific application what modality you may want to use when considering things like spatial resolution, sensitivity, and cost. And so with that, I will send it back over to Tanya, and we will answer any questions that you might have.
Excellent. Thanks again, both Katie and Ayaz, for the presentations. And now we do want to take a few minutes to answer some of the questions that have come through. They're fantastic. And take a look at some of the questions that we have here. So one of the questions that came through is probably for Katie. Is it possible to integrate, for example, optical imaging with other imaging modalities such as CT or X-ray, similar to how you would with PET imaging and have multimodal imaging?
Yeah, of course. So it's definitely possible. So there's many systems out there that have actually integrated these right into the system. So you can have, and most of them already have both bioluminescence and fluorescence capabilities, but they have systems that are integrated with either X-ray or CT. And again, that's just a way to get that both data sets within the same system. But I would say with optical, because it is typically a 2D modality, you do have lots of options to, use other complementary modalities, even though they're not in the same system. So for example, you could combine it with MR or even PET if you wanted to do that, even if it's not in the same system. But yeah, I would say it's similar to PET, where you can definitely have those options to integrate.
Perfect, excellent. And so we talked there about multimodal imaging. You had spent a bit of time talking about multiplexed imaging. I hear people use them almost interchangeably. Are you able to kind of just, I guess, clearly define the difference between the two?
Yeah, so they are often used interchangeably, which makes it a little confusing. Multiplex, I would say the important thing there is that your signals are coming from the same medium. And so I mentioned for, let's say for bioluminescence, this would be you'd be using two different bioluminescent reporters to track different things. Or for PET, you'd be using PET and looking at two different PET reporters. That's what we would refer to as multiplex imaging. You can also have multimodal imaging, where you are still tracking two different, let's say, biological processes within the same animal, but you're using different systems. So maybe you have a bioluminescence reporter, and then you also have a PET reporter to look at something else. That would be more considered multimodal. It gets a little bit confusing because people can also say simultaneous multimodal. So that would be something where we have an integrated system such as PET MR. And I would say that would be multimodal rather than multiplex.
Great, excellent, thanks. I'm going to take a couple of questions that will be directed at AES and the endomicroscopy system. And so the first one, I'm going to kind of have three different questions here. But when using the endomicroscopy system, does the animal need to be sacrificed at the end of that study, or can the system be used for longitudinal studies?
So it depends what organ are you imaging. So if you can make a small suture and like small incision and reach to your desired organ, like let's say liver or kidneys or intestine, then there is an option to have a sterile sheath. So this instrument comes with a sterile sheath that goes on the top of the probe, and then the sheath is sterile. So then you can go inside the body of the animal, do the imaging, take out the probe, make a like suture 1 or 2 stitches, and then you can do the longitudinal imaging. But if you are imaging brain, or if you are imaging, like let's say lungs, then the incision would be like really damaging to the animal. So in those cases, like if you can work it around, it's fine. But then in those cases, I think it would be difficult to do longitudinal imaging. So it depends upon your tissue. The instrument has the capability of longitudinal imaging.
Great. So speaking specifically about the brain, when imaging the brain with the system or this type of imaging modality, would you be able to do it by creating a cranial window? And obviously the skull would need to be removed to access the brain, but if you were able to place a cranial window, could this type of technology be used?
Yeah, so what I have done in the past, so I think I went to Dalhousie University, where there's a neuroscientist who is working with this kind of system. So what we did was we did not remove the skull completely, rather we shaved the skull down to like very thin bone. So it's not like the skull is completely exposed or taken out, but then still there is a fine bone between the skull and the probe. And then we imaged the brain, and the images were captured fine.
And so how deeply below the probe surface can you acquire an image?
So you can go, theoretically, you can go up to 400 micrometer, but because bone has like really thick optical density, so I think we could go up to 180 something.
Sorry, you said 180?
180, yeah.
Got it, thank you. And then continuing along the line of different organs, how does this type of imaging work on things that are moving, such as the lungs or the heart?
Yeah, so the frame rate, the rodent hearts, they beat very fast. So it is not possible to image like the beating heart. So one cannot expect like, yeah, I can look at the live beating heart, but then you can do ex vivo imaging for the heart. So basically you take the heart out of the tissue and then flash freeze it or put it in the stain, and then you can image it. But beating heart is, the system cannot image the beating heart because the frame rate is slower compared to the rodent heart beating rate. Lungs can be imaged depending upon which part of the lung that you are looking at. So if you are rightly looking in the middle of the lung where the movement, the motion artifact is going to be really high, then it's going to be difficult. But if you are looking at the tip, then it's really interesting. So we are trying to develop a protocol with UHN at Toronto, and then we are trying to look at the lungs ex vivo. So this group takes the lungs out and then puts them in the organ chamber, and there they are trying to mimic the function of the lung. And there this probe can be placed, and then you can see how well it can be imaged. But unfortunately, because of this COVID situation, those experiments are on hold. So once we reopen, we might get that results and we will see how it performs.
Perfect, excellent. I'm going to take a couple more questions that are more directed at Katie. Does the type of mouse used for a study need to be considered when choosing a modality? And further to that, are there advantages to using an albino mice versus other strains when doing optical imaging?
Yeah, so that's a great question. I would say the mouse model definitely needs to be considered, especially for optical, because again, you're relying on the light getting out of that tissue. And so the fur can definitely have an effect. And you may have noticed in the images, we actually typically, especially for if you're using a mouse with black fur, we will shave or nair the area of interest just to, get as much light out as we can. I always recommend to people, if you can, if your model allows it, try and use a nude mouse model or something without hair, just so that you don't have to worry about it, especially because with optical, you have that nice benefit of being able to visualize the whole body. And so using a nude mouse allows you to see everything. But if you don't have that option, you can really use any mouse model. Just again, if you can, try and get rid of the fur as best you can in that area of interest.
Can I
Got
add
it.
on to that?
I just wanted, sorry, AS, to cover one really
Mhm.
quick question, and we'll get back to people and expand the answers as needed. So one final question before we just reach the top of the hour. Katie, you talked about ex vivo imaging, and is it possible to do this with bioluminescence imaging? And if so, and expanding on what you'd already said before, how does doing ex vivo imaging really help to improve the issues of limited depth of penetration with optical imaging technologies?
Yep. So bioluminescence is very similar to fluorescence in terms of you are able to do ex vivo imaging. You do need to remember, depending on the luciferase that you choose, as I mentioned, for some of them, you do need cofactors such as ATP and oxygen. And so that gets a little bit complicated because as soon as you sacrifice the animal and take out those organs, you have a limited time frame to be able to image them ex vivo. Now, it's not as tight as you would think. You typically, you have enough of those cofactors. You usually have about an hour window, but you just need to, navigate that timeline depending on your model, because you want to make sure that you're administering the substrate before sacrifice, giving it a couple minutes to circulate throughout the body, getting to those cells of interest, and then you would sacrifice your animal, and right away, you need to get those organs out. And that can be really great, as I mentioned, to better localize the signal, because now you're the signal is going through much fewer layers of tissue to be able to get that light out. So that can really help if you have a lower detection limit.
Excellent. So we have reached the top of the hour, and I do want to be respectful of everyone's time. There are a lot of other fantastic questions, and we will capture them all. We'll create a Q&A transcript, and later today or tomorrow, you'll receive a link back to the registration page, actually, and you'll see that that's been updated with the recording of today's webinar, as well as the slide deck. And once we get the Q&A finished, which may take us a few days, that link will also be provided there. So right now I'd like to thank Katie and Ayaz for their great presentation. I do hope that we were able to provide you with some information and insights into the different imaging modalities and how they may help you with your studies. If in the days and weeks to come you have additional questions that come up as you're thinking about your research and what we discussed today, I do encourage you to reach out, and I'm happy to put you in touch with Katie or Ayaz directly so we can talk a little bit more about the science that you're doing and how these imaging modalities may help you. I also encourage you to join the live demonstration of the systems that we're going to be doing next week. Again, we'll send a link to that in the follow-up email that you'll receive this afternoon or tomorrow. And we look forward to seeing you at another Scintica event in the near future. And thanks again for your time today.